because we have access to biotechnologies that allow us to do this large-scale perturbations. So here are some illustration of the perturbations I'm going to be talking about today. So these are enabled by gene editing tools, where you can see you can go into each individual single cell and make very precise edits on individual single genes or combinations of them. And after perturbing those genes in the single cells, you can then read off their corresponding effect using different technologies. For example, on the top is something called perturb-seq, whereas at the output, what you will see is this big cell-by-gene matrix, where individual entry here correspond to individual expression of individual genes in corresponding cells. And at the bottom is something called the optical full screen, whereas after you perturb the genes, you can read off their effect by doing this types of high throughput imaging, where you can look at phenotypes of what's happening, like what are the shell shapes and what are the different kind of places that the molecules go. Just to give some intuition about the scale and costs of such experiments, we have access to genome-wide data, which correspond to perturbing every single individual genes, around 20,000. So you have 20,000 different perturbations, and you can read off the effect of those perturbations in single cells, either using perturb-seq or optical full screen. But the cost for such genome-wide experiment for perturb-seq is quite expensive. It's more than $1 million. For optical full screen, it's relatively cheaper, but still not to the extent where you can do this type of genome-wide screen in every different context, in every different disease. So the question we're asking is, can we actually develop computational methods that can substitute some of the experiments by predicting the effect of unseen perturbations? So intuitively, you can think of this being that I have a context of interest. I perhaps targeted, say, 100 different genes, as illustrated by like different arrows there. And I want to know what's going to happen to the 20,000 minus the 100 that I haven't tested on. So this problem has been acknowledged to be quite difficult. This paper called GEARS is one of the first few deep learning-based approaches for solving this question using gene ontology and a graph neural network. But then after 2 years of this being published, there's a paper that says those deep learning methods, including GEARS and some others, actually does not yet quite outperform very simple linear baselines. And then even more recently, there's a paper called Systema that basically discover if you look at control cells. Yeah. If you look at the control cells and look at what those different perturbations are doing to this group of control cells, you see that actually different perturbations lead to very similar directions. So using this observation, you can come up with a very simple baseline. So practically, if you just use the mean average of all the perturbations you have seen to predict the unseen perturbation, in fact, it's probably going to do pretty well. And they benchmark this against a lot of different approaches out there, including GEARS as well as more recent foundation model approaches. And you see the purple line here actually to be outperforming those more complicated methods. And that purple line is just predicting the mean of the training perturbations. So this has been a quite hard problem, mainly because you're kind of doing an out-of-distribution generalization where you've never seen the data coming from the unseen perturbations, and it's unclear how to predict it. But today I will show you that we can make some progress by learning generalizable causal insights from existing data sets. And then upon having a predictive perturbation modeling, I will show you that it can be used to guide future dataset collection more efficiently. So concretely, I'm going to talk about 3 parts. The first one, I'll spend some time on basically the theoretical and algorithmic basis to learn causality from data. And this is going to lead into an algorithm that predicts the effect of unseen perturbations in the second part. And then I'll just quickly show you an application of using this model to design the next perturbational experiments. And these are the list of papers I'm going to cover. So I'm gonna go into the first part, just to give some intuition of what it means by causality behind data. Here's a gene regulatory networks that people use to represent causation behind a certain process in sea urchin. The point of having this diagram here is to show you that they're actually highly complex, where individual nodes here correspond to genes, and then you can see the directions as being regulations of one gene's effect on another. So if we actually think about computational methods to achieve something like this, we can identify very quickly two different limitations. The first one is basically if you run an existing method to discover a network like that, you would run into problems with low accuracy and high computational costs as the graph size gets larger, as the number of variables in your system gets larger. So here's running a PC algorithm, a canonical causal discovery algorithm in a simulation. As the number of variables increases, you can see the execution times climbs up. This is log scale, and then you can see the accuracy actually drops. So that's the first limitation. I'm going to spend like maybe 2 minutes on it. And then I'll quickly jump into the second limitation, which is actually trying to deal with this high throughput data. When we look at data like this, we would be wondering like, what are the actual units we want? Maybe in a gene regulatory network that I just showed you, what are the units between which we want to learn the causal effects between? Just like being very apparent in this image of the cell, you cannot take individual dimensions here because they're just pixels, and you don't want to learn a network between pixels. So you have to learn the causal variables first before actually discovering what the network between them are. So I'm just gonna spend like 2 minutes on the first limitation to just talk about what the problem of causal structure learning is and show you our result. Of course, happy to talk about it afterwards, and you can also find more details about it in the paper. So essentially, the approach we tackle, the direction we tackle the first problem is trying to minimize the number of conditional independence tests. I'm going to tell you what conditional independence tests mean. So the problem of causal structure learning is essentially you are given data sampled according to an unknown causal graph. And what you want to do is learn the causal graph from the data sampled from it. And you can do so by just running conditional independence tests in the data that you have access to, because certain graph structure actually details conditional independencies in the data. The problem with the prior work is they actually use a number of conditional independence tests that's exponential in the maximum degree of the graph, which you can imagine as your graph size grows larger and as it grows maybe denser, D is going to be quickly unmanageable. So what we did in our work, in a nutshell, is we showed a lower bound of exponential in S number of tests required for any algorithm to solve this problem. And then we also develop an optimal algorithm that uses this many of conditional independence tests, where S is the maximum undirectly click size of the graph. Just to show you maybe some example of how these two bounds compare to each other. So these are the papers. In reality, you might have often the case where the maximum degree is pretty large, whereas the click size is small. This is because clicks, they correspond to fully connected hubs in the graph. And then if I take a gene regulatory network, this one from the literature, the maximum degree is actually on the order of 1500, around that. And then the click size is on the order of around 12. So by reducing the bound, we can perform less conditional independence tests, and you can imagine that this is going to be more computationally efficient and also more accurate because you're running less number of tests and you require less assumptions. Okay, so that's the first limitation. I just want to talk about this for just this 2 minutes. And I'm going to the second limitation, which is the case where we have high dimensional data like this. What you see here, this matrix, it actually contains about 20,000 to 30,000 different columns. So you have gene expression measurements between those numbers. As you imagine, if you want to learn maybe a network between those number of genes, it's going to be probably requiring a lot of samples and maybe not so useful. So our proposal here is instead maybe some of the genes, as you see from the matrix here, they have very similar behaviors across different cells. So maybe they have very similar functions and should be grouped together as one causal unit. And once we learn those causal units, we can then learn what the network between them looks like and interpret the network between different groups of genes. So essentially, with the data here, there are different rows here. And I was describing early on, we have this type of perturbation data, which means there are groups of cells coming out from the observational distribution, which are just the control cells where no perturbations was applied to them. And then there's also going to be samples coming from perturbing individual genes. Like groups here correspond to perturbations on gene I1 and all the way to perturbations on gene IK. And since we want to learn groupings of genes that are also going to be perturbed individually here, you can imagine this is something that will come out of the whole thing, where we are grouping certain groups of genes in which you can see the coloring of the bars, they're being perturbed in those different conditions into one single variable here, x2, and then maybe some of the other genes, they correspond to the other groups of causal variables here. And we want to learn basically the groupings of those genes as well as the network between the groupings of those genes.
Oh.
Yeah.
So you want to find the two similar genes, but how to define the similar? What is the similarity function?
What is the similar function?
Between two genes.
So intuitively, they would have very similar. So intuitively, they would have very similar behavior across the single cells. So you can imagine like being just two columns here that have very similar expressions across single cells. So they co-express. But then mathematically, I'm gonna introduce the definition, if that's okay. Okay. So mathematically, what happens is the data we have access to are observational and perturbational data sets annotated by OI1 up to OIK, whereas the observational cell O is a function of latent variable X, where X is sampled from the control distribution. And each individual perturbation data set, OIK, is going to be the same mapping function F, but of the causal variables that has been perturbed. So the XIK under those perturbational data sets would be sampled from PIK. And PIK has only very limited constraint change from P, where it has to be the case that the perturbed the perturbed dimensions, the effect only propagate downstream. And then this F function is shared between the control and the perturbed distribution. And then to answer your question, what are the two genes going to be in this case would be I have IK and IK prime, and they correspond to the same, basically the same latent variable X. So their effects can be explained by just modulating the same causal variable, and they're sharing the same mapping function F to the output space. And then intuitive, what it looks like is going to be just they co-express in the output space.
So you just calculate electric distance between the two latent variables as their similarity function, as their similarity score.
Maybe that's a simple approach that you can do. Yeah, but theoretically, this is the definition of them.
Yeah.
I had a question.
Yeah.
I understand now how you're measuring similarity between, say, the two profiles that you represent in yellow.
Mm-hmm.
But there's also these arrows that you've defined, which articulate some notion of causal relationship.
Yes.
But I'm not sure how that translates to what you're showing on the right.
What I'm showing on the right. Yeah, that's a good point. So what that shows up on the right is the definition of PIK compared to P is going to be that IK only changes the mechanism of its targeted variable in the graph. So what I haven't given here is just the definition of joint distribution P, which is going to factorize according to the graph structure. So P is, in this example, is a joint distribution between x1 to x4, and that's going to factorize according to this graph. And then PIK would also factorize according to the same graph, but only the mechanism of IK is changed. So that's a hidden point. I haven't put out the details on the slide yet, if that makes sense.
It does. Thank you.
Thanks. Okay, I'm gonna move on now. So when you look at this assumed data generating process, the question we're asking in the first place is, given this data generating process where we only have access to samples like OOI1 up to OIK, can we learn a unique latent model that explains the data? Or in other words, can we actually identify something about F, P, and PIK? So a precursor of this problem is in the case of without any perturbational data, where you only have access to O. And then the precursor assumes an empty graph between the latent variables. So all of them are independent. And that's known in the literature as the independent component analysis, where basically you have independent latent variables that generates your observed O. And people have been solving this, and there are quite a lot of literature on this. And there you can see the hardness of the problem, which is even in that empty graph case, you can have multiple pairs of latent F and X that generates the same observational distribution O. Because you can imagine that you can have a multivariate Gaussian X, and you can just randomly rotate the Gaussian vectors and absorb that rotation into the mapping function F, and then it's still going to get you the same, basically observed O's. But with perturbational data sets, especially like this ones where we have individual genes perturbed, we can actually show a much stronger identifiability results. I'm not going to spend too much time on it, but just to tell you the result is that if we have certain assumptions on the number of perturbations we have access to, as well as that the causal effects are not canceled out, which is known as faithfulness, and then some constraint class of this mapping F, we can actually identify just what are the targets of this I1 up to IK. So we know which genes correspond to the same latent variable, and we can also identify their structure between them. Of course, up to permutations, but this doesn't really cause too much problem. So that's the theoretical result. I'm gonna now move on to maybe simpler cases where I introduce the algorithm of how to learn the perturbation effects. So if I have lost you, you can maybe come back now. So to develop an algorithm to actually learn maybe causal representations and the effect of perturbations, we can look from just the data perspectives. So here's what we have access to. So we have access to a group of control cells and also groups of perturb cells. And we might have access to multiple different perturbations. So you can imagine that I have this illustration of the red arrows, and I maybe have like another groups of cells coming from like yellow arrows and green arrows, so on and so forth. So to learn the changes of perturbation that's due into the control cell, we can take any type of feed-forward neural network. And here's just an example of a variational autoencoder. I don't know why it's not showing the gray box, but there's supposed to be a box that's like, now you're seeing just this very wild, empty, combined representation. There's supposed to be a gray box. But anyways, so you can take any feed-forward network, and then you can input here, O is one control cell. And then you can also input what the perturbation you want to generate the perturb cells are corresponding to. And then you can combine their representation and decode to actually a perturb cell. So taking this here, you can input, this is what's happening. And then essentially the perturb encoder just takes in maybe just an indicator, say, for example, one hot encoding that's telling you that I'm now generating perturb cell correspond to the same perturbation. So while doing this, we can generate maybe a bunch of perturb cells correspond to the perturb version of the control cells. But the problem is most of the technology that I was telling you before to measure the cells, they're actually destructive to the cell. So once you measure it, you kill the cell. For most of the technologies so far has been like this. So that entails the fact that you will never actually observe one cell before and after perturbation. So now you generated perturb version of my control cells, but I don't know individually how to compare one single perturb cell to the actual group of perturb cell that I sampled, because and they're just essentially unpaired. But to solve this is actually also very simple. What we can do is we just input a batch of control cell and generate a batch of perturb cell. Although we don't know in the individual pairing, we know that the distribution of this batch of cells should look like the distribution of the actual perturb cell. So that's what we're doing here. So instead of training with a very standard MSE loss, we just do a distribution loss between the batch of perturb cell to the ground truth perturb cell. And in this case, we're taking maximum mean discrepancy because that's quite easy to compute and also doesn't really suffer finite estimations of curse of dimensionality. So this is the reason we're calling the framework a discrepancy-based approach. And you can solve this VAE with any feed-forward or generative models if you like. I will just take VAE because that's, I think we're sufficient to solve the expression problem here. Okay, just 1 minute on how this connects to the theoretical result. We can actually split open this combined representation. So what happens is we encode a control cell to latent X, and we encode a corresponding perturbation. And then as long as we're forcing the combination, we're forcing the perturbation effect to follow some kind of causal graph here, then we can get a perturbed version of the representation that reflects this network. So when we learn this whole thing together, we can actually prove identifiability, which is with infinite data, minimizing the training loss, actually achieves the theoretical identifiability detailed above before. Of course, you have to satisfy also the assumptions there. So that's just 2 minutes on how it connects to the previous result. And then what can we do with it once we have this network trained? I want to say a few words. So one thing you can do with it is you can perform predictions in terms of combinatorial perturbation effects. So what I mean by that is you might have, as in this illustration, perturbations on, like, say, gene 1 on a group of single cells, and then perturbation on gene 2 on a group of cells. And now we're interested in, say, I perform just combinations of perturbations of both gene 1 and gene 2 on the same cell, what the effect will look like. And you can do this in a zero-shot manner after you train the model with just the single perturbation effect. So what happens is you just input both I1 and I2 to the perturb encoder, and then you encode both their effects, and then you propagate basically their effect to downstream variables to get the perturbed version of X1 and I1 and I2. So the theoretical basis of this is basically in the literature of causality, the definition of combinatorial perturbation is very compositional. So the definition of perturbing both I1 and I2 will have this sort of factorization where the effect of I1 and I2, they're like compositional. So you have already seen what PI1 and PI2 looks like in their corresponding single perturbations. So now you can just use that to predict what's going to happen to the cells if you do both of them. Of course, if the definition change, the theoretical fit basis doesn't really sustain. So we've also tested this on a combinatorial perturb-seq on cancer cells from a 2019 paper. And you see here, these are UMaps where individual dots correspond to cells projected down to 2 dimensions. And then the gray blobs are actually all of the cells in the data set. And then you see the actual cells from the combinatorial perturbations are in blue. And then the combined prediction from the zero-shot manner are in orange. They quite overlap very well. But of course, as I said before, if the basis of the definition change, for example, you have interactions, then this doesn't necessarily guarantee you to predict the effect perfectly. So that's for combinatorial perturbation effects. But then what this framework cannot solve is if I were interested in, say, for example, perturb gene I1 and I2, and now I have unseen perturbation on a novel gene I3, what's going to happen here? And the main reason is because the perturb encoder essentially we're feeding in just like one hot encoding to indicate cells coming from the same perturbation. And if it's a non-seen single gene perturbation that has never been seen during training, this is going to be a one hot encoding that has never been seen during training. And of course, this encoder is not going to map it to any meaningful latent effect on the latent variables. So that's the thing I will try to solve in the second part, which goes into this paper on bioRxiv. The solution to this is also very, very simple. So instead of just using a one-hot encoding, we can just use a representation of the perturbation from prior knowledge base. You can imagine that I have gene I1, gene I2, I3, all kind of studied in prior literature, and I can extract the corresponding embeddings of those genes from prior literature. And this embedding for the unseen perturbation is going to be only similar to those in training if those genes have been seen in the prior literature to have very similar effects. And once you have this very reasonable embedding for the unseen perturbation, you can then try to generalize to its effect. So we call this whole thing a modular framework for predicting responses to perturbational changes, because essentially you can use any prior knowledge base here, and then you can swap out encoder, decoder, or whatever you like. So just a few more words about what I mean by prior biological knowledge. So here are 4 examples of prior biological knowledge you can use. The first one is you can use, essentially, you have this control cells already in your training data, which are cells with no perturbation applied to them. But you have essentially measured all the individual genes across those control cells. So to represent perturbations on any gene, you can just take the corresponding effect, take their corresponding expressions across the cells. So if you use this, essentially, the unseen gene perturbation will have a similar embedding to the seen genes perturbation if they always co-express together. So that's the first approach. And then the second one is you can take those pre-trained single cell foundation model and essentially get gene embeddings from those pre-trained single cell foundation model, which captures a lot of information about how single cell and genes behave in maybe different observational situations. And then the third option is you can take, say, like a language model like GPT, and essentially you can input just text description about those genes. And this paper, GPT provided different options. Like you can input NCBI description or gene card description about the genes into the language model, and then just get the embedding from the second to last year or something like that from language model, which basically summarize prior literature information about those genes that this models has been trained on. So that's the third option. And then the last option is you can take maybe other type of data sets measured about genes, like this dependency map. And this essentially came out of the Broad Institute many years ago, where they provided viability score, which correspond to if you knock out genes in different cancer cell lines, what the survival rate of those cell lines are going to look like. And you can just take the columns here, which correspond to the gene's viability across those cancer cell lines as a representation for that gene. So I sometimes ask people which one of the priors they think is going to work the best. Anyone want to try it? To just guess which one of the 4 type of prior knowledge would work the best?
Tax
embedding. Any other answers? So actually, I think tax embedding works quite well. So this is one test we've done on genome-wide screen on cancer cells. So you see that this is measurements of distribution loss, MMD. So lower the better. It's measuring how the predicted perturb distribution look like the ground truth perturb distribution. And the text embeddings are in between here, GPT, GPT, so on and so forth. And you see that they do quite well compared to using single cell foundation models or just very simple baseline of using the control gene expression. But actually, DAPMAP in this case works a little better because this is essentially also measuring perturbation effects in similar cancer contexts. So not quite far anyways. And there are actually more priors explored in a concurrent work called Presage from Genentech that you can read about. Okay, going back to our work, essentially I've shown you how those prior representation compare to each other, but what I haven't answered is, does the whole framework actually works better than all the different baselines I told you about before, like using the very simple prediction of just the training perturbations mean. And that corresponds to just essentially this orange bar here. And you see there's 3 different metrics here, MMD, RMSE, and retrieval rank. So these 2 are lower the better, and this is higher the better. And you can see that more using that map feature actually performs better than those baselines. And GEARS is the first deep learning approach using gene ontology type of prior with the graph neural network. And then this one is the linear method, but we changed their corresponding embedding to DAPMAP because DAPMAP actually works better than the ones they provided before. And this is the simple baseline. And you can see the performance compared to them. Anyways, there are more benchmarks in the paper that you can read about. How am I doing on time?
20 minutes.
Okay.
Mostly.
Sounds good. So essentially, MORPH is, I think the major contribution of its performance gain is coming from this type of MMD training using single cell level information. If you use, say, like RMSE or randomly paired cells, you probably are going to do pretty worse on this MMD type of metrics. And then secondly, it's going to perform well compared to some of the methods because it can use this very informative prior biological knowledge to relate unseen ones to those ones seen during training. And I just want to maybe show another example of having this modular architecture. So not only you can use various different prior embedding here, you could also swap the input to the encoder and the output to the decoder with various different modalities. So the ones I've shown you before are using these omic features, which is pretty simple. You have just this tabular data sets. But you can also use imaging features that's coming out from this cheaper type of measurements like optical pole screens. And once you extract the imaging features, put it through MORPH, it can just give you the predicted perturb features of the same modality. So concretely, I'll just give an example of how we apply this to one optical pole screen on HeLa cells when they have been infected with Ebola virus. So this is what the data look like. You have images of single cells. And then this is circling out one single cell out here. And there are various different channels in this optical pole screen, including DAPI channels. You can see the nucleus here. And then VP35 RNA. VP35 is something very of interest to Ebola virus infection. And you can see also the protein localization of VP35. So what they also provided is for each individual single cell, classification of which infection stage those cells are in. So including faint, punctate, cytoplastic, and peripheral. So as input to, so we're interested in this type of basically classification of infection stages, which is explain the reason why we're doing the follow-thing to extract the image features. So what we did here is we use a pre-trained vision transformer And we take it here and then fine tune it on basically the classification of the infection stages, which then gives us a kind of image feature for each individual single cell that reflects the infection stage. And you can see a UMAP plot of the imaging features looking like here. Each dot is still one single cell, but they're computed based on the imaging feature extracted from the vision transformer. And it's colored by just basically their infection stages. You can see this is well clustered, which means the representation is likely going to be very informative of the infection stage. So this is essentially what we take as input to morph. And what we did is just train morph on a subset of perturbations. So each perturbation, essentially, you can look at just where the perturb cells lie in this UMAP. And for this MPC1, you can see all the blue dots. That's where the single cells from perturbing this gene correspond to. So most of it is in terms of faint and punctate, whereas non-targeting is spread all over the place. So what we did is we take we withheld a subset of perturbations out from training, and we try to test if morph can predict what the proportions of these different stages those unseen perturbation will lie in. And that's measured by just the cumulative mean L1 loss here. And you can see that morph in the green bar actually corresponds to much lower loss compared to the baselines, and especially just on the perturbations where their effect is most obvious as compared to the non-targeting. Whereas if you move to the perturbations where they essentially don't do much changes to the proportion of different stages, the non-targeting, as well as the other baselines are not so different. That's one example of applying morph to predict unseen perturbation effects in optical full screen. And with the ability to predict perturbation effect, you can actually use it for various different downstream tasks. Like you can do transfer into new cell lines. You can do gene interaction prediction. You can also try to read off the gene regulatory network by just poking at the latent representations. But what I will show you in the last part is an example of using this perturbation modeling for the design of perturbations in next round. So as a warm-up, I will show you a semi-synthetic simulation of doing target nomination, still on the Ebola virus optical full screen case. And this is a semi-synthetic simulation in the sense that we're just trying to mimic a real experiment and see what it does. What we did here is we took 3,000 perturbations as training, and we withheld essentially 15,000 perturbations without seeing their actual effect. And we use morph to train on the 3,000 and to see if we can predict which ones out of the 15,000s are the ones that are likely to lead into maybe earlier infection stages. So this is a plot where a tertiary plot showing you where those 15,000 perturbations lie in terms of their proportions of faint, punctate, cytoplastic plus peripheral. And this objective is just adding faint and punctate. And I'm annotating here the top 10 targets predicted by morph, just trained on the 3,000. And you can see that some of the targets predicted to be top by morph are actually just ground truth very top here, having very high objectives. In particular, this are RAB RAMS 7A, CCDC 115. These ones predicted to be top 10 by morph, just trained on 3,000, are actually just top 10 in the ground truth ranking. So this is just a semi-synthetic simulation showing how this can be applied. And now I will show you an actual application we did in collaboration with Near Hokoten's lab at the Broad. So essentially, this is the experimental pipeline. We're interested in nominating targets that could potentially be helpful for cancer immunotherapy. So the experiments started by having just a data set where our collaborator tested the perturbations on 73 target genes that they deem to be probably helpful for the downstream application. So this is a data set where we have 73 single gene perturbations, a lot of single cells coming from it, and we can see their effect. So after perturbing the immune cells with those perturbations, they inject those perturbed cells into a mouse with melanoma cancer, and then waited 2 weeks to just read off the effect. And after reading out the effect, we also annotate the cell states of those effects. And you can see 5 different colors here. Some of it correspond to progenitor states, which are stronger at cancer killing capability, and they're also terminal exhausted state. So these are still like single cells, and you can imagine that each one of those 73 single gene perturbation is going to be like just a subset of the single cells, and that's going to have like a various different proportion lying in those different cell states. What we actually wanted is a model that can predict the cell state proportion coming from the ones that we haven't tested in the 73. We want to see if we can use this model to identify better targets that bias the proportion towards progenitor. So I'll just show you directly the result. You see here, again, it's a tertiary plot. And to read it, you can just take each single dot and do like a projection perpendicular line to each one of the axes. And then this is going to basically show you the proportion of progenitor, and this is cycling, and then this is terminal exhausted. So here, the dots correspond to just the targets, and the green ones are the ones that they selected in the expert selected screen of 73 targets. And then you can see 2 black dots here. These are non-targeting perturbations. So you can imagine that if you just like maybe randomly select a target, it's probably going to look something like that. And then the red dots are the ones that are predicted to be leading to more potential for cancer immunotherapy by the machine learning models. And this is coming out of a result of an actual experimental validation. And the coloring here, I should have explained it earlier, this is a checkpoint blockade therapy derived objective, so higher the better. So you see overall here, if you compare the green dots to the red dots, you see that the green dots, they actually do quite well. So a lot of them lie at places with higher objective than the red dots, which means they are very good at selecting targets. So the prior knowledge already cover a good enough space. But luckily, we were able to find these 2 targets that actually lead to better objectives, which gives us some confidence in terms of what the machine learning models can do. Although they're not perfect, but we might still be able to get something out from it. Also, I want to comment on just comparing different methods in terms of predicting the perturbation changes. So in the cancer cell cases, I've shown you this plot before, and you can see that how different biological priors compare. And then in the immune case as well, we also benchmark a lot of different methods of presenting the genes. And there's one that's called perturb T cells, which is essentially taking all the literatures from prior literature that perturb the T cells in one way or another in various different applications. And you see in both cases, the strongest prior to represent the gene actually came from previously collected perturbational data sets. So they might have been a cheaper modality like measuring viability, or they might have been in a different context, like in this case. But the strongest prior always came from just similarly perturbing those genes. Which means that if we actually want to maybe perfect the perturbation predictions, this is one way we can think of utilizing iterative collected perturbational data sets. And I just want to maybe say that in the case of considering iteratively collecting perturbation data sets to update the model, another axis we want to consider is in terms of how to use the model and existing data to select the next batch of experiments. So I've shown you before, very easy when we have 2 rounds, we just do maximizing the objective. But if you have multiple rounds, there might be other things you need to consider. And then something in the literature called acquisition function is just estimating the optimality of adding data from new experiments in the next round for a particular objective. And we have this early paper in the case where if we're interested in obtaining a desired state, that might be very rare in the output space, which is often the case of finding perturbation leading to a particular rare state in the biological case. We can actually use extreme event theory, like rare event theory, by using just the inverse probability of the occurrence for that perturbation in the acquisition function. Okay, and I just want to end by saying that the community, I think, is pushing towards perfecting perturbation prediction for virtual cell and other objectives. And at the Broad at Eric and Wendy Schmidt Center, we have this cell perturbation prediction challenge. Which is essentially just a website hosting different challenges that we've done in various disease contexts where we collaborate with wet lab biologists to design a setting to have machine learning algorithm built. And then they're going to test in the lab of the top performing methods to see if your algorithm actually works in the wet lab setting. So this website will be, I think, alive soon, not yet alive. We have hosted 2 different challenges already. One is the cancer immuno machine learning competition, which is essentially just the experimental design I told you about. And there's, I think, almost closing the obesity machine learning competition that just happened a few months ago, where we're tackling metabolic disease. And I think there will be one new competition coming out later this year, so stay tuned. And yeah, at the end, I'd like to acknowledge the PS I've worked with during my PhD and also my wonderful collaborators and student mentees. Thank you all for listening. I'm happy to take other questions if there are any. Thanks.
Yeah. Students. Anywhere here or online? Yeah.
You mentioned when you were talking about the theory that I think it was like for single perturbations, it was recoverable, the latent graph, like of the permutations. And since you're doing this with very like VAEs, you impose any structure in the network for symmetry or something to deal with the fact that it's recoverable up to perturbations, or do you not need to worry about that?
So I think we do have some parametric assumptions. The parametric assumption is the parametric assumption for the theory to work is the mapping function from the latent to output has to be a certain set of polynomials. But the latent distribution, they actually can be arbitrary. Any like non-parametric assumptions of structural causal graph can work. But when we do it in the VAE, essentially the VAE has, when we initialize the exogenous noise to be random Gaussian, that's like an additional assumption on the exogenous noise to be Gaussian. But then still the structure can be anything that can be observed into just the decoder and encoder. I think that was one set of assumptions I can remember, but I might be missing something. So I think the paper's appendix has a lot more information about that. Yeah, it's from a few years ago.
I'll do a question online. Any comments on how does this scale to non-viral disease?
Non-viral disease?
Yeah.
How does the basically optical pulse weighing scale?
Like I'm not sure, like scale to non-viral diseases.
So I think as long as the wet lab have figured out a way to culture the cells and perform single gene perturbations and measure their effect, and then we maybe have like a few hundred single gene perturbations, we can probably do an okay job of predicting covering the space. But that only has to exist when they have figured out just in the wet lab how to do this.
Training so those gene regulatory network
Mhm.
X1, you have one arrow like towards X2 and
Mhm.
X4. So those are the unknown or the know like knowledge for your training.
So I mean,
Do you use that?
yeah, that's a good question. So, sorry, I'm gonna give you an extended answer. Short answer is in the ones that are presented so far, you don't need it. So when you don't have it, you can still train it, and you can extract it afterwards. But then your question might be targeting when I have a network existing known, maybe I should be using this in the model, right? And you can use it in very different ways. You can use the gene regulatory network, you can use the graph neural network to extract corresponding prior gene embedding from the network. And that's one way this network can be incorporated. And another way is you constrain just some kind of latent embedding to follow that path. And that's another way to incorporate it. We have not benchmarked this thoroughly, but my guess is the first type of approach would work better by extracting some prior information from, extracting some type of gene embedding that is reflected of the network might work better than constraining the latent space of the network. Yeah.
Did you mention the Arc Institute, their state paper?
The state paper, I didn't mention that. Yeah.
Can you briefly share like what's the difference?
Oh, yeah. So okay, so to my understanding of to my understanding of the history of the paper is okay. Well, we wrote the NeurIPS paper, the theory paper, that was using MMD for training single gene perturbation effects. And then later on, we were doing this more paper, and then at the same time, I think Arc Institute is doing the state paper, and they were using like a similar kind of training objective of mapping control cells to perturb cells. Yeah, they cite me in I think the method section of doing that. Although I might not be the first one to use MMD for training such single cell type of models because there's cell OT before, which is using chemical perturbations. But anyways, and then state majorly focused on, I think, transferring of perturbation effect in different cell lines. So if you read their paper, you will notice that they actually use like all of the existing perturbation data sets out there to train like one big model, whereas we're doing more like kind of a targeted setting of like having one single context. How can we make this very single context work better? So I think if you're just like particularly interested in one single context, maybe using morph is better for that. But there's like they have like a lot of work trying to benchmark across different cell lines and something like that. And you can see there, yeah. And we also have like maybe an extended discussion of how to read off the gene regulatory networks and how to actually apply this to optical pole screen. And I think they did it majorly, all of it on transcriptomic data.